Ever wondered how traits are passed down from parents to offspring? How do we inherit our eye color, height, or even our predisposition to certain diseases? Welcome to the intriguing world of genetics, the science that seeks to answer these questions and more. Genetics, the study of heredity and variation in living organisms, is a field teeming with discoveries that have shaped our understanding of life itself. So, buckle up as we embark on this fascinating journey through the history and development of genetics. Our journey begins in the 19th century with a humble Austrian monk named Gregor Mendel. Mendel, curious about the nature of inheritance, turned to the garden pea plant for answers. Over the course of 7 years, he meticulously bred and crossbred these plants, observing the traits that were passed from one generation to the next. Through these experiments, Mendel discovered patterns that revealed the basic principles of inheritance. He formulated what we now know as the laws of segregation and independent assortment, cornerstones of Mendelian genetics. The law of segregation states that each parent contributes one of 2 gene copies to its offspring. The law of independent assortment, on the other hand, states that different genes sort into gametes independently of one another. These laws, though simple in their essence, were revolutionary. Mendel's laws of inheritance laid the foundation for the field of genetics. Fast forward to the 20th century, the mystery of heredity starts to unravel. The year is 1953, and 4 scientists are about to make a groundbreaking discovery. James Watson and Francis Crick, with crucial contributions from Rosalind Franklin and Maurice Wilkins, unveil the structure of DNA, the molecule of inheritance. This deoxyribonucleic acid, or DNA, is a double helix, a twisted ladder, if you will. It's made up of 2 strands of nucleotides, which are molecules that contain a sugar, a phosphate group, and a nitrogenous base. These nucleotides are arranged in sequences known as genes, and it's these genes that hold the instructions for life as we know it. But how do these instructions make sense? Well, it's all about the language of life, the genetic code. This code is written in a sequence of 4 different nitrogenous bases: adenine, thymine, cytosine, and guanine, abbreviated as A, T, C, and G. These bases pair up in a specific way, A with T and C with G. It's this pairing that forms the rungs of our DNA ladder and encodes the instructions for building and maintaining an organism. With the discovery and understanding of DNA, the language of life was finally decoded. The blueprint of life was laid bare, marking a monumental milestone in the field of genetics. Now, armed with the knowledge of DNA and its structure, scientists embarked on understanding how this blueprint works. The DNA molecule, with its spiraling double helix and unique nucleotide sequences, was not just a static structure. It was a dynamic entity, constantly involved in the intricate dance of life. One of the most fundamental processes in this dance is DNA replication. This is a complex mechanism where the DNA molecule duplicates itself, ensuring that each new cell receives an exact copy of the genetic information. This replication process is a marvel of precision, with enzymes and proteins working in harmony to unwind the DNA helix, copy the nucleotide sequences, and reassemble the strands into 2 new DNA molecules. But DNA doesn't just replicate, it also communicates. This communication takes place through a process called gene expression. In gene expression, the information encoded in DNA is used to create proteins, the workhorses of the cell. This involves 2 main stages: transcription, where a segment of DNA is copied into a molecule called messenger RNA, and translation, where this messenger RNA is read by structures called ribosomes to assemble proteins. And these proteins, my friends, are the building blocks of life, involved in everything from cell structure to immune response. Yet life is not static, and neither is the genetic code. Genetic variation is a key aspect of genetics, providing the raw material for evolution. This variation arises from mechanisms such as mutation, where changes in the DNA sequence occur, and genetic recombination, where different genes can combine in new ways during sexual reproduction. These processes, along with environmental influences, contribute to the stunning diversity of life forms on our planet. Understanding these processes of DNA replication, gene expression, and genetic variation is essential to grasping the big picture of genetics. It's like having the keys to the library of life, where each book is a different species and each word a unique gene. The processes of DNA replication, gene expression and genetic variation are fundamental to understanding life and its diversity. As we move into the 21st century, genetics takes a monumental leap forward. The Human Genome Project, a massive international collaboration, embarked on a mission to map and sequence the entire human genome. This unprecedented endeavor, completed in 2003, shed light on our genetic makeup, revolutionizing our understanding of heredity and disease. The revelations from the Human Genome Project have far-reaching applications across various fields. In medicine, genetic testing can identify individuals at risk of inherited diseases, leading to early intervention and personalized treatment strategies. In agriculture, genetic engineering can enhance crop resilience and productivity, promising a more sustainable future. And in forensics, DNA profiling has become an invaluable tool in criminal investigation and paternity testing. Yet the power to read and manipulate our genetic code isn't without its ethical quandaries. Who owns genetic information? How do we protect privacy when our most personal data, our DNA, becomes accessible? And how do we ensure that the benefits of genetic research are distributed equitably without exacerbating social inequalities? The Human Genome Project opened a new frontier in genetics, but with it came a host of ethical questions that challenge us to use this knowledge responsibly. As we look towards the future, the field of genetics holds immense promise and challenges. The advent of gene editing technologies like CRISPR-Cas9 has opened up possibilities that were once only the stuff of science fiction. Imagine being able to accurately cut and paste genes to cure genetic diseases or to engineer crops that can withstand climate change. But it's not just about editing genes. The future of genetics is also about personalized medicine. As we better understand our genetic makeup, treatments can be tailored to individuals, increasing their effectiveness and reducing side effects. This is particularly promising in the field of oncology, where genetic insights can guide the development of targeted cancer therapies. Synthetic biology, too, is an exciting frontier. By designing and constructing new biological parts, devices and systems, we can create everything from biofuels to disease-fighting bacteria. Such innovations could fundamentally change how we interact with the natural world. Yet, with these advancements come significant ethical considerations. That's where genetic counseling comes in. Genetic counselors play a vital role in helping individuals and families understand and cope with genetic risks and conditions. They provide support, education and guidance, helping people navigate the complex landscape of genetic information. The future of genetics is boundless, promising exciting breakthroughs that could revolutionize our understanding of life and our place in the natural world.